All right, everybody. Today we are going to go over the week 4 review, which is going to cover chapters 13 and 17. These are the last chapters that are going to be on the midterm exam. So finally, I don't know, I'll get a little bit of breather after this one. All right, starting with chapter 13. What is an infection versus a pathological state or disease? An infection is just whenever you have colonization of microbes in a tissue or an area, typically where they don't belong or they don't exist there normally in that number. So pathological state or disease, both of those are referring to, we have presentation now of some sort of deviation from health. So presentation of negative symptoms that would be representative of a deviation from health. Okay. What are endogenous versus exogenous infections? Endogenous infections are infections that are caused by microbes that came from within your own body. We had talked about the best example. I just think it's just the best example, so I keep bringing it up. But is whenever you get a UTI, it's typically caused by E. coli from your own colon. So it doesn't belong there and it causes bad symptoms. Then we have exogenous infections. These are caused from any sort of microbes or pathogens that are outside of the body, like COVID, for example. So an opportunistic pathogen versus a true pathogen. An opportunistic pathogen is something that can become pathogenic outside of its normal sort of situation. Whereas a true pathogen, that's something that is always a pathogen. Like that's just what it does. It's like COVID, right? That's always a pathogen. It's not opportunistic. It didn't do something else separate. So we think about the brain eating amoeba and how horrible it is. It has a 97% mortality rate and all that. And it kills so quickly they can't even treat you. Well, that's terrible, but that's not what it normally does. It doesn't normally invade people's sinuses and cause brain eating disease. That's not its usual MO, so that would be an opportunistic pathogen. A virulence factor is anything, geez, there's a gnat in here, sorry. A virulence factor is anything that gives a pathogen an advantage in colonizing or creating illness within a host. So the basic example, the go-to is always going to be capsules. So capsules make it hard for phagocytes, which are some of our white cells that go around like sentry cells, eating up bad guys, eating up pathogens. It's hard for them to eat up those bad guys whenever those bad guys have capsules on them. They're sticky, they're hard for them to get purchase on. Okay, an infectious dose. Infectious dose is the amount of microbes that it takes to cause an infection, to establish an infection in a host. So that could be as little as few as 1. There are diseases that only require 1, but there are some diseases that require 50 to 100 microbes to be present in order to establish infection. So that's infectious dose. The steps involved in establishing an infection. First, we have to have entry. So we talk a little bit in this chapter about portal of entry. So that could be through the respiratory tract, it could be through the skin, through like puncture of the skin. It could be through drinking or eating and all sorts of things like that. So that's our entry aspect. Then attaching to the host tissues in order to establish, an infection in that area. Then we have so the capsule helps with that, right? So would Fimbriae, and then viruses, of course, have their spikes that interact with the host glycoproteins. That's important to remember. The host surface glycoproteins on the host cells are what the spikes interact with in order to adhere or adsorb onto the host cell and then get ingested by the host cell. But anyway, so those are some ways that pathogens can attach. Then you have to survive the host's immune response. Some of these organisms, like I said, they have capsules to evade that immune response, or they could even produce toxins that could kill things, or things, if we were talking about bacterium, things that could digest, penicillin that was in the area caused by nearby mold, or from drugs that we're taking. All of that stuff is going to be included within being able to survive that host immune response. But some of those are going to be, most of those actually are going to be considered virulence factors as well. So there's quite a few things that would be considered virulence factors. So let's not forget that. It's not just a list of like 5 or 6 things. It's pretty much anything that the bacteria would do that helps it live. All right. After surviving the host onslaught of immune response, then we would be causing disease. So that's usually causing damage to the host or having the host cause damage to it themselves, which we'll get about in a moment. And then exit, some portal of exit of that virus. So whether you are, vomiting and that leads to spread, or you're coughing or sneezing like you would with COVID and that leads to spread, or not washing your hands, all of this stuff would be included in that exit sort of part of things. So just wish I would mark these off as I went, so it'd be way easier to see where I am. Part 8. Which causes more damage, typically the microbe to the host or the host to themselves indirectly? The truth of the matter is it's usually the host to themselves. And why would you cause damage to your own self? Well, you're trying to cause damage to the microbes, but your immune response is causing damage to your own cells in the meantime, because you're just like, I don't even care. Let's just, get this sucker out of here. We're just going to do what we can to kill it. And so the immune system does that, but it also causes inflammation and damage of cells nearby. And that's usually what leads to development of certain symptoms associated with a lot of these diseases. And that's the truth of it. Of course, there are microbes that will cause damage themselves, whether they are producing toxins or what have you, but the post response has a huge, huge hand in that. All right, signs, symptoms, and syndromes. Signs are anything in an infection that can be measured. That includes anything from palpable swollen lymph nodes to fever that we can take with the temperature for it, to white blood cell count. Then we have symptoms. Those are things that are described by the patient that we can't necessarily measure. Things like pain and discomfort and fatigue and nausea, those sorts of things. And then syndromes are basically diseases that consist of a set of symptoms and signs that we attribute to that disease, but it can be caused by a number of microbes. So you have a headache, you're photosensitive, sensitive to light, your neck is sore and you're tired, and maybe have a fever. And all of those are symptoms of meningitis. But what causes meningitis? Well, it's not any one thing, especially because you can have bacterial meningitis, you can have fungal meningitis, you can have viral meningitis. So what is causing it? It could be a number of bacteria or viruses or even the fungi. So yeah, you would need a lot more testing than that, but they would all be diagnosed as meningitis. So meningitis in that case would be considered a syndrome. This is true for pneumonia as well, and the cold. If you have a cold, oh my gosh, that can be caused by so many different viruses, like hundreds, literally. Okay. Leukocytosis versus leukopenia. Leukocytosis is increase in white blood cells. Leukopenia is a decrease in your white blood cells. So we have septicemia versus bacteremia. Bacteremia is the presence of any bacteria in the blood, not necessarily flourishing, but just the presence of bacteria in blood. There should not be any there. And then septicemia is the flourishing of the bacteria in the blood. So they're growing and multiplying within the bloodstream. So latency, remind me again about latency. Latency is referring to getting infected by a virus at face value or any other, it could be other microbes as well. It doesn't have to be a virus. So we can even see this in tuberculosis, which is caused by a bacterium. But these pathogens come in and they establish an infection within a host, and then they kind of lie dormant and just stay there and wait for changes in the immune system to allow them to want to come back out and redo their thing. So that we see that in tuberculosis, and we see that in herpes. We see that in a lot of things, actually. So that's latency. And what are sequelae? Sequelae are things that are lasting effects that have occurred because of an infection. But even now, even though the infection has been cleared since then. So when we see FDR in the wheelchair, that was due to symptoms, sequelae that were left from having had polio, right? So that is an example of that. people who have had COVID, some of them have lasting effects as well. So what are the 4 phases of infection? The 4 phases of infection, essentially talking about progression of infection here. And that's going to start with incubation. So no growth there, just like in the bacterial growth curve where we have the log and the lag and all that. So the lag phase in bacteria is equivalent to the overall picture of infection for incubation. Then we have the prodromal phase. The prodromal phase is where we start seeing the very first symptoms associated with a disease, signs and symptoms associated with the disease, very first ones. Then we have the invasion phase. The invasion phase is where we are full on multiplying at our maximum, full on causing disease and everything like that. And then we have convalescence, which is like recovery from the disease. Reservoirs versus carriers. Reservoirs are beings where pathogens are can grow within them, usually causing no or low level disease, usually with some sort of symbiosis or something like that. And then a carrier is somebody who has gotten the disease, maybe doesn't have any symptoms and is carrying it from having been exposed, maybe even from a reservoir, and now carrying that into the population. So the carrier is not somebody who is, technically a reservoir for this disease. They are just a temporary, like almost like a vehicle for the disease, right? So fomite and a nosocomial infection. Fomite is a vehicle for transmission. So like this pen, if I were to lick it and then hand it to you, and then you get my COVID or whatever it is, then this would have been a fomite. It doesn't inherently have any infection or anything. It is just going to get exposed to infection briefly and become a fomite, a vehicle for the transmission of that disease temporarily. Then we have nosocomial infections. Nosocomial is just referring to healthcare related infections. It's just another term for that. Okay, compare and contrast prevalence and incidence, morbidity rate versus mortality rate. So this kind of takes a little bit of breakdown just because of the words, but prevalence is talking about the frequency of a disease, like how common that disease is. Literally, you can write it down in numbers, like this many cases over time, right? So prevalence is referring to the frequency of that disease, basically overall. Whereas incidence, you're looking at a specific time. So how frequent that disease was this year. Okay, where that usually is the same thing as morbidity. Here we're talking about the presence of symptoms within that population and the frequency of those symptoms within that population related to that disease, but they are essentially the same thing, morbidity and incidence. Then we have mortality rate, which is the death rate from that disease. Epidemic versus endemic versus sporadic versus pandemic. An epidemic is anytime you have an outbreak in a region where the disease is at numbers that greatly exceed, and there's ways that they measure this specifically, but where the numbers of occurrence of a disease, any disease, greatly exceed what is normally seen in that region or population. So anytime we have an outbreak like that, that would be an epidemic. Then we have endemic. This is whenever a disease has established itself within a population. It is not strange to see, at least at those numbers. Sporadic would be referring to random outbreaks. You keep hearing me talk about rabies. Those are sporadic outbreaks, right? Within the human population, it's very sporadic. And then we have pandemic, which is referring to spread of disease across continents. So that's it for chapter 13. Let's move into chapter 17. Chapter 17 talked about diagnosing infections, and this is a brief one. So where should you start when diagnosing a patient's illness? You should always start with the patient's face value symptoms, right? So it's talking about their symptoms, talking about their signs, start there, and then move on after that to things like blood work and tests and things like that. So always starting at that face value with the signs and symptoms. What is the difference between presumptive data and confirmatory data? Well, the real breakdown of it just happens to be that presumptive data will get you as far as like the genus identification of the organism, whereas confirmatory data is going to get you as far as the species identification of that organism. What role can biochemical testing often have to play in diagnostics? Well, a lot of organisms, especially when we're talking about bacteria, can be separated from one another based on their ability to metabolize certain ingredients or create certain byproducts from metabolizing these ingredients. And that can cause color changes in the media that they're growing on and allow us to identify basically the species and such like that based on that, based on their metabolic response to the media. So if we don't have available to us serological tests, if we don't, like with a known antibody or a known antigen associated with that disease, or we don't have primers for us to do the PCR, then what the heck are we going to do? You could run biochemical tests in order to separate this out. Man, if we're talking about the plague, right, because we do have that in the United States, that would be a great example, right? So we don't always have stuff available at your hospital that would allow you to run these tests, but you can get answers still and use things like a dichotomous key, which is our next question. A dichotomous key, and I'm actually going to get one out just so that I can show you because I have some in this thing. Cabinet, I guess is the word most people will use for this. Okay. This is a dichotomous key. And so you would start over here where you are gram negative, gram negative. And let's say you did a test to determine, is it, does it ferment glucose? Does it produce gas whenever it does it? No, it doesn't produce gas. Well, then you move on to the next question. You want to know, and same thing, but for sucrose and acid production. So no acid and sucrose in that case. So you'd move on to this one. So that would be doing a nitrate test. Do they reduce nitrate? Yes or no? And then based on that, let's say they do. Was there acid in that glucose test or not? And then you would be able to say if there was, then it would be Providencia alcalifaciens there. So that's how a dichotomous key works. It's just doing biochemical tests and following that down to try to determine the identity of your organism. So hold on, let me put this away. Okay. Anyways, so we did dichotomous key and how it would be used. You do your tests, your metabolic tests, and then you would use the data that you collected from those tests in order to determine what kind of organism it could be. And you can use something that's quite similar to that, might not be a direct dichotomous key, but we have Berge's manual. And Berge's manual is used in a similar way in order for us to identify bacteria based on all sorts of characteristics. So this is a slanted form. Okay, PCR and how is it used in the medical laboratory? You guys heard me talking about this in the lab. I'm glad that I did bring it up. But remember, PCR, polymerase chain reaction, it amplifies DNA. That's what it does. That's its base purpose. So it can take a very, very small, minute amount of DNA and amplify it and give us billions of copies in a matter of just a few hours. And each cycle that runs, and you might have to calculate this, so that's why I'm bringing this up. Each cycle that runs, you go from one double strand to two double strands because we split this apart and then you make a copy of it. And so these two, you will like split this one apart and you make two copies from that. So on and so forth. So sort of an exponential growth in number of the strands that are going on for each cycle that's going on. That's how PCR works, and we use it as a diagnostic tool in a laboratory. We can use it to make comparisons of DNA as well as identify microbes, as long as we do have the primers associated with that to identify those microbes. So that's how that would be used. The two functional uses of Kirby-Bauer assay is another one that I mentioned just briefly, but it does have two functional uses. One of them is to identify usable antibiotics to treat a patient, and the other one is to use it for identification of that microbe. If you're not sure what it is and how it reacts to this, could be part of that key And you could use it as part of that dichotomous key route in order to find the answer for what this organism is. What is serology? Serology is whenever we do testing from serum, a patient's serum, and often this is related to immunology. An antibody is a protein that is produced by someone in response to an antigen, and an antigen is anything that your body, your immune system can respond to as foreign. So that antibody is that protein, and Y-shaped little protein. And whenever we are trying to confirm a diagnosis of somebody, we might want to see if they have the antibody that they're producing against COVID, for example. And if they have the antibody against COVID, that means they probably have COVID. And if we want to know the antigen, do they have the antigen for COVID? Well, we would have to have a known antibody, in order to test for that unknown of whether there's an antigen or not. So we did talk about this a bit whenever we were talking about ELISA. Remember the indirect ELISA? Indirect ELISA is whenever we are trying to test for the presence of the antibody, and direct ELISA is whenever we're trying to test for the presence of the antigen. And it's also the sandwich method. So, all right, what is a Western blot and what is it usually used for? Western blots are just, we run a protein out on a gel, transfer that protein onto a membrane, and then expose it to certain antibodies to see whether or not a protein or another antibody could be there. And it's often used for verifying the results of what we get for an ELISA. Specificity and sensitivity. Is one preferred over the other? No, one is not preferred over the other necessarily. We would like both of them at the maximum at all times, please, but it doesn't always work that way. We'll sacrifice one or the other depending on what the test is and what we are testing for. So specificity, here we're talking about a situation where we only bind to the target thing. That will eliminate false positives, but it'll risk false negatives, because if we're only binding to the target thing and not anything else, we might sometimes not bind the one that's the target. So that's going to risk false negatives. Selectivity is we will test positive even when there's only a very minute amount of the target present. So we'll find it. It's there somewhere. If it's there, we'll see it. This will eliminate false negatives, but it does risk false positives. So that's why we would want both if possible. So that covers the review for chapter 13 and 17. Everybody, seriously, good luck on the exams. I'll see you guys for the review, the true reviews for the lecture and the lab soon. So everybody take care. I gotta figure out how to stop this.